Have you ever wondered how unicellular bacteria are able to infect and cause disease in large multicellular hosts like us, putting up a fight against complex and specialized immune systems? How do they do it on their own? The truth is that they actually don't. Despite being independent organisms, bacterial cells work together. Cells often form biofilms and release virulence factors as a group. But this raises another question. How do they know if other cells are around them and react to that? This is all part of an amazing mechanism called quorum sensing. Quorum sensing is a system for bacterial cells to detect the population densities of bacteria around them. They then use that information to decide whether to act as individual cells or as part of a group. Let's take a look at how this works. The first step of quorum sensing is the production of autoinducers, which are specialized signaling particles that are released by the cell into the environment. Different autoinducers have different roles in bacterial cell functions. Since nearly all bacteria release these molecules, they are strong indicators of cellular density. If the density of bacteria in an area is high, the autoinducer concentration will also be high, since there are more cells releasing them. On the other hand, when there is a lower bacterial cell density, there will be fewer cells producing autoinducers, leading to a lower autoinducer concentration. Some autoinducers are species-specific, while others can be recognized across different types of bacteria. Autoinducers in the environment are detected by receptors. These receptors exist on the surface of gram-positive cells and in the cytoplasm of gram-negative cells. After binding to an autoinducer, either the receptor complex phosphorylates another compound that then becomes a transcription factor, or the receptor complex becomes a transcription factor itself. Again, this mechanism differs between gram-positive and gram-negative cells. Ultimately, the resulting transcription factor binds to the cell's DNA, either activating or repressing transcription. This generally results in the expression of genes for group functions, like biofilm formation and toxin release, while suppressing genes for motility and other independent functions. A really interesting example of quorum sensing is found in Vibrio fischeri, a type of bioluminescent bacteria that lives in the ocean. The bacteria's bioluminescent effect is controlled by quorum sensing. The cells will only glow if they are part of a larger group, but why? The reason for this is that bioluminescence is an energy-expensive process that isn't worth doing if it doesn't pose some sort of benefit. If the bacteria were floating freely in the ocean as individual cells, there would be no point in wasting energy on bioluminescence since that won't help the bacteria in any way. However, Vibrio fischeri often live within Hawaiian bobtail squids as part of a symbiotic relationship. The squid provides a favorable environment within its body for the bacteria to live, and the bacteria makes the squid glow, which blocks its shadow in the ocean and allows it to blend in with its environment more easily, making it less noticeable to predators. In this case, the bacterial cells are living as a large group. Bioluminescence is helpful to the host and in turn the bacteria itself. Quorum sensing shows how bacteria aren't just isolated cells. They work together to grow and thrive as a community. Thank you for watching.